Thank you very much, Dr. Bill Hafez, who is generous and kind, and you and the Society of Emirates Society of International Medicine is honoring me to give to you this lecture. I'm especially pleased, and I saw the poster, and I was especially pleased the young students who are participating, and you are cordially welcome. I trained at least, and I'm not bragging, then I am honored by at least 250 MD, PhD students, and they went all out of the world. One of them is here in Abu Dhabi, Dr. Dana Haddad. She was my student. She's a breast cancer surgeon here. Okay, before we got too much in the past, I just would like to say that the research which I talked to you about and therapy is going back 230 years, and I will show you why. And then we are using a drug which was used especially in children, vaccinating against smallpox for 250 years, and all the clinical trials, safety trials, and everything were done. And now, hopefully, we will be able to convert it to a cancer vaccine. So I start and showing to you, so I am the person here, and I just would like to declare that I started based on our own research. I wanted to say, I talk today about our own discovery, nobody else's. Not to neglect others, but just to show how it came. So I started a company, company in 2002, and the name of this company is Genelux Corporation, Dr. Vale referred to. And this company, I do not have anything to do since 2014. This company now just went on NASDAQ and working on virus usage in cancer therapy. The second company I started 2 weeks after I resigned from the first one, and or 2 months, and then this is STEM Immune, and this company I left 2021, and after that I started the third one, Immunolox, which is still working, and I'm working on it. So this is virus alone, this is stem cells and virus therapy, and the third one is virus and immune cell therapy. All right, so roughly 30 years ago, I used to be a faculty member at Cornell. We started with light-emitting proteins. I worked a lot on light-emitting protein, luciferases, GMP, lighting up living systems. And there, we had bacteria which we injected to tumorous mice with tumors, and you can see that, lo and behold, not the whole mouse lit up, only the tumor. And we questioned then, could we eventually use the bacteria as tumor targeting, and hopefully, if we engineer them, tumor killing agents? But also, we asked the question, yes, and this is how the image shows how the bacteria grow in the tumor really long time. It was in a glioblastoma tumor. And then we questioned, but do we need to engineer bacterium, or do we have one living killer which can multiply only if it is inside of the cells? And then we selected Vaccinia virus. It is a cowpox virus. It is not a human pathogen. And this Vaccinia virus was used by Professor Jenner in London, 1796, 230 years ago, first time, to inject this young boy to protect him from human pox. So the drug was discovered and used, as I said earlier, for 200 some years, happily, in 250 to 500 million children, and mothers brought their children for vaccination protection. Nobody really appreciates what it meant that all the clinical trials and all the FDA and everything what we are facing with, we did it naturally, and mothers took their children for safety. So now, if I continue to start, we also designed very early on that if we take the genome of the Vaccinia virus and introduce a light-emitting gene, this virus becomes like a flashlight or a light-emitting system. Wherever the virus goes and multiplies, photons are coming out, and we can detect it. And this was a very original construct. We published that in Cancer Research early on, I think 2007. Then we also had to know if we introduce something into the virus, do we interfere with the life cycle of the virus? And luckily, we did not interfere with anything, and we will see that this is a virus particle, and this is a DNA of the virus, and this virus is sitting inside of our cells, but does not interfere with our nuclear genome. So we do not need to worry that we genetically alter the human genome, because this virus never interacts with our human genome at all. So we have a cell-infecting virus replicating in the cytoplasm and lysis, killing the cell if it is replicating. Now, when I do not want to be now specific, this virus has its own RNA polymerase, and this RNA polymerase is making many, many copies of the virus protein. It has over close to 200 messenger RNAs and protein, and lives that way. And it was, we just published this one in Cell. I do not want to bore you with it. But one important thing is what I wanted to show you is, funny-wise, in this RNA polymerase, we have a tRNA. It is a glutamine tRNA. We do not know what the role is as part of the RNA polymerase. But the most important aspect is that tumor cells have lots of glutamine. So it seems to be that the sensor, God or nature, built into the RNA polymerase of Vaccinia, which is sends it and letting initiate it. So the next slide shows more details. I do not want to talk about that at all. And show now how to look the glioma tumor, put on the mouse, and inject it with Vaccinia intravenously. And you can see on this slide that only the tumor is glowing and the mouse is not glowing at all. So we were really lucky, because we have the tumor-killing agents and goes to the tumor only and not somewhere else. It is also very important that all our cancer therapy agents today are killing also the non-tumorous cells in us. In this case, luckily, we found something that, at least in mice, and I will show you later in humans, kills only the tumor cells and not the healthy cells. And you can see here the green fluorescent protein, which we engineered into the virus, and it shows in the human tumor on the mouse that only the tumor tissue is glowing. For example, the arteries and the blood vessels are not glowing, so the mouse tissue is not affected here at all. And when we went on now and we did our first real tumor regression experiments, and it was human breast cancer in mice. And you can see that doesn't matter which route you are using, tail vein, femoral vein, or intravenously, you can see that after 28 days, the tumor is gone. The non-injected mouse tumor is continuing to grow and killing the mice. So practically, and here we can also show the imaging data, how the virus is in the tumor, and the cross-reacting material, and finally the tumor is cleared. So it also shows that how the amount of the viral particles or killers are located in where, and you can see that in the tumor we have 10 to the 9 particle, somewhere in the circulation 100. So it is an enormous enhancement, we also question now it is only valid for one cancer or many. It works also beautifully, and you can see here how mesothelioma is completely cleared up in most tissues. So just very shortly, the principle: we take the virus, inject into the bloodstream. The virus finds the tumor cells, infects them, and perhaps I will show you later, activates the immune cells of the body and eats up the tumor, and the tumor is gone, and hopefully never comes back again. Vaccination. We also asked whether only solid tumors, or we can work also with metastasis, and it works with metastasis also beautiful. And you can see here, for example, lung surface metastases are completely cleared up and done. And so once more, Vaccinia virus replicates and eliminates also circulating tumor cells. The circulating tumor cells are the more toxic part of oncology. After the solid tumors, metastases are growing in us, then also the circulating tumor cells, which are finally giving the patients the last heavy burden. So, and also we can also identify, I do not want to go into the markers and the biomarkers, but we can also identify from the blood and the circulating tumor cells based on lighting up the infected cells. So we asked also only solid tumors or liquid cancers can be also attacked. And if you look at this slide, it shows that also leukemia, B cells are infected and aggressive disease. You can light it up beautifully as a diagnosis. And the same thing here with monocytic leukemia. And I'm just going not to waste too much time of Dr. Gale's program fast. Now, we asked the question. Now, I mentioned it originally that if we engineer bacteria, would this bacteria deliver a toxin? But now the question is here, can we engineer, in addition to the lysis activity of the virus alone, some drugs or some other components into the virus, and can we use our tumor as a drug factory? And as long as the tumor is producing the drug, as long tumor cells are, the cells are producing the drug, and they are killing the rest of the cancer. So can we use our own tumor as a fermenter for drug production? And we put in here Avastin from Roche, and this slide shows here the insert, and you can see that the non-infected tumor grows. The Avastin inhibits, but if we combine it alone, if we combine it with Vaccinia virus, Vaccinia virus, Avastin is X-fold better. I went to Roche 15 years ago and showed them this slide, and I said that, look at, Avastin is fantastic. helps many tumor patients or cancer patients inhibiting blood vessels, but look at what it does in combination with Vaccinia. We also asked, can we use it also for imaging? And we put in the sodium iodine symporter, and the sodium iodine symporter here shows you, again, it's part of Vaccinia. Dana Haddad is my graduate student who did this work. She's here in Dubai, breast cancer surgeon, but she could not come, she is ill. And she did these experiments and in Germany, and you can see that we can use enhanced PET, positive emission tomography imaging, because the virus enhances uptake of iodine. But not only allows imaging and PET imaging, it also allows the treatment of triple negative breast cancer, because if we have the virus and the virus is gathering lots of iodine radiation, then it helps to kill triple negative breast cancer. This we published together with Memorial Sloan Kettering with Yuman Fong, who is a colleague of mine. And so on, you can see that the triple negative breast cancer with the HNIS in the virus and the non-treated tumors are growing just with iodine that way as controls. So now there were the preclinical data just they showed. The virus recognizes tumor cells and kills them. The virus recognizes tumors, metastases, and circulating tumor cells and kills them. The question was, and healthy cells are not harmed. The question was, can we now go to FDA and can get protocols, and what can we do with FDA's careful supervision or watch? And so we also had a major advantage that as you would like to use Vaccinia virus, it has to be GMP manufactured. The GMP manufacturing, I saw here one of the lectures will be perhaps about that, is very important. Without the GMP approved criteria, you cannot put anything into humans. It is obvious to you. So we started clinical trials after GMP manufacturing in several clinics. There are the lists here, and we did also different routes. I do not want to go into any details, but I think it is very important that in ovarian cancer patients and in the peritoneal virus replication, we could already cure the 10-liter ascites with just a simple injection into the abdomen. So 10-liter ascites, nearly 95% of the tumor cells were eliminated, but it was an enormous success. Even the clinicians didn't want to believe that it works that way. So it is also now in clinical trials. We also could take out the infected cells from the human body, and this is re-isolated, I think, from breast cancer station, and this one is from mesothelioma, this is from Kettering, and also we are able to use different markers and even identify it in the virus infectivity. This slide I like the most among our 30 years work. This is the first slide when we showed in London, Royal Marsden Hospital, a patient laying in his bed and a lesion in the darkened room could be lit up by just turning off the light, and I sent them from San Diego's detection camera system. And it was done based on light emission from the engineered virus, and it was done 2013. When I went next year and gave a talk in Cambridge, an American clinic director stood up and somewhat vulgar sounding, told me when he saw that, Professor Szalay, how the hell did you put into humans GFP? I wrote 10 applications and FDA. I told him, sir, perhaps you didn't realize I did not put in into humans GFP. I put in GFP into the virus, and the virus carried into the thing. But I like this one very much. It's a beautiful story. All right, so there are many considerations for the human trials. I do not want to dwell on you. And also, most importantly, you need many, many good young people, clinical associates, graduate students, MD, PhD students, and also cancer patients. So I just made a list here for memory. So Genelac was the first company. This virus applied virus alone treatment. So nothing else, just a naked virus. And there are other conditions. They started out with ovarian cancer, and this ovarian cancer are the platinum-resistant cancers. And this is now, I am really proud to say, because I was no one who ever had any clinical experience, and now it is in phase 3 trial, and will gain approval, hopefully, by FDA in less than half a year. And I'm humbled that I could help our female population and could eliminate an enormous suffer all over. But I don't have anything to do with the clinical trials. So as soon I transferred it to the company, I had to step back, and they are informing me like every other shareholder. But they need money, of course, and they need that. So important is that you see now phase 3 is now finished. The slides were done earlier, and hopefully next, not later than next June, the approval will come. It is already fast-tracked and so on. Yeah. And we have the primary endpoints and so on. So phase 2 trial data were that good that 80 clinics in the United States applied to the company to be allowed to do phase 3 trials. So data were very, very good. So then we also said, now how is it with stem cells? What will this virus do with stem cells? You know, many, I think in one of the program after me will be stem cell therapy. So we took stem cells and added the red virus to green stem cells, and you can see that, lo and behold, the virus infects also mesenchymal stem cells. So theoretically, mesenchymal stem cells could be also the carrier. And we also looked at different induced pluripotent stem cells. And when you see here the red, it is a red virus infecting induced pluripotent stem cell. So theoretically, we could take induced pluripotent stem cell, liver-specific, heart-specific, muscle-specific stem cells, and add back to the patient his or her own stem cells, like really to convert any type of or prevent any type of rejection. So now even the stem cells can be used as a carrier, and we did these experiments, and I will show you the problem which we are all facing, cell transport people. We have autologous stem cells and allogeneic. There are my Trojan horses, who are stem cells who are hiding the virus, and they are both originally from Greek. Some of the inventors into the company brought the stem cell horses basically. So we have Trojan horses. So first we started out with stem cell extraction from adipose tissue. And the adipose tissue is, of course, available in everyone, and I used to say, especially to graduate students, don't go to the gym because you might throw away very, very valuable resource for future cancer therapy or something. But this is a simple method. So a ping-pong paddle size of anesthesis is given to the back, 50 ml fat is taken, the fat is separated from the cells, and 1 hour later can be injected to the cancer patient either intravenously or intratumorally. So this is being done. And now Dr. Lander, I think he has some contacts already to Dubai to bring this machine. He can put this extracted fat into this machine and can produce large amount of GMP level stem cells. So it is already moving directly from patient in the laboratory into the GMP manufacturing stem cells. So, and it can be done in the GMP. So the adipose stromal vascular fraction has several very, very important components. People using it for joint injection, anti-inflammatory but here we are using the stem cells infected with Vaccinia, and it's working very well. And also it is going to certain type of the virus infects from this fraction certain type of cells. And also what we see is that the stem cells, if you mix it with a virus, the blood cannot kill the virus. So it has a protective effect for things. It was all unknown due to the light emission. We could see that all of a sudden the red virus grew in the presence of stem cell. But most importantly, I just wanted to show this is a mouse experiment. And in this mouse experiment, you can see if you intravenously inject the stem cell plus virus, you see an enormous protection with a stem cell. If you do it with intratumoral injection, nearly everything. So it is very important to take the incoming virus, very, very important, and protect it. So now I am saying that in Dubai or in the Arab Emirates with an enormous humbleness. Now it turns out that every one of our countries in the world have Vaccinia virus because we are not vaccinating against smallpox. So every country has the stockpile. And the stockpile is very important. It's like gold made already, and we are waiting for biological warfare to protect ourselves from the bad people's pox. This is very important. But at the same time, every country has a drug ready to treat cancer. We tried out 10 different countries' virus preparations, and every one of them, doesn't matter it was manufactured in Beijing or in England or in the United States or in Soviet Union, former Soviet Union, all the Soviet countries has this Soviet virus and so on, is killing cancer. So I asked already Dr. Vale when I was here last year that we should find out that in here, whose, perhaps you own, Vaccinia virus do you have? And we could try it out, it works like that, and clinical trials can be immediately started. You have the tumor patients, you have the virus, and we have the permits in the United States to use the virus in clinical trial studies. So, sorry, I just wanted to point out that this is the American vaccine, and this American vaccine is now at CDC and the military. And the military, when I called up the German military, what is a German vaccine, or what was a German vaccine? Could you send me some, because I heard that you have also stockpile. You know what they said? No, sir. we discarded it. And I called up the president of the company, and I said, I was told by the manufacturer that you have vaccine. He said, yes, sir, but we wanted to find out what are you planning to use it for. So you might face the same answer we got in Israel. So you might have also a protective answer here in the Arab Emirates. But important is you have the vaccine already. I just want to let you know. And GMP. So we worked also a lot on autologous versus allogeneic system, and here now we are using the different stem cells, and we are using intracranially injected virus-infected cells, glioblastoma, and you can see that the peritoneal blood contains virus. Most importantly, the peritoneal blood has an enormous enhancement of central memory T cells, which goals that we are doing not only elimination of the thing, but vaccination against the cancer. So I just wanted to show you, and not to be inappropriate, but it's a medical community, 2 patients with the adipose tissue-derived stem cells infected with Vaccinia. The first patient is a 68 years old male, and he had the tumor size, thyroid cancer size. Here we injected after 30 days, the tumor started to inflame. 65 days is here, and 85 days the tumor is completely gone. It was 7.5 years ago. The patient is still tumor-free. I'm not saying it braggingly, I'm saying it humbly and thankfully that it seems to work. The next slide will be even more drastic. This is a head and neck cancer patient, and this head and neck cancer patient had that large cancers that he could not tumor, that he could not sleep anymore. Mayo Clinic, MD Anderson, everybody rejected him, and we injected the patient, and you can see that after half a year, tumor is completely gone, and the patient is alive now again 70. We have an 96-year-old prostate cancer patient. He was also stage 4, and we injected him, and he is now 97, 98, and perfectly well, no symptoms of prostate. So this was while I was a president of the company, and after I left, of course, the company is doing all FDA approved, controlled clinical trials. Now the phase 3 trials are being finished. So therefore, I am not talking about the company. I cannot, it is not my duty, but this is going very well. As of to date, I think in the 2 trials, or 2 to 3 trials, over 1,000 patients were already treated. So this was when I wrote the slide that time, initial efficacy is obtained, and ACAM 2000, the American pox, is working. So now this one was the second company with a glioblastoma trial. I do not want to go. We are using into surgery, after surgery, into the brain of the patients, and they are working, and there are the people who do the clinical trials as well as the preclinical data. The last company I started, and I'm finished, last company I started 2021, while I was always, and I am always, university professor. This company is intellect-wise, perhaps it is not very, very novel in a sense, because now instead of semi-immune, I made Immunolux, so Genelux, I made Immunolux. But this company is now taking T cells, immune cells, and try to see what can we do with immune cells and Vaccinia. And lo and behold, we are very lucky. We started to do with CAR T cells and infected CAR T cells with Vaccinia, and the CAR T cells work 5 to 10 times better than without Vaccinia. And I do not want to bore you with, we characterize everything, and you can see that the CAR T Vaccinia combination is very red here. Oh, sorry, Jesus. God, no, I don't. Can you take me back to the last 2 slides, or how can I do? I pushed the wrong button. We have to wait now. And the CAR T cells are, here. Thank you. The CAR T cells are CAR T cell Vaccinia combinations infecting, and we did glioblastoma as well as neuroblastoma, pediatric versus adult cancers, and seems to work. So my secret hope is that adding Vaccinia to CAR T, solid tumors can be also treated. Now, as you know, leukemias are the targets for CAR T. So this is to CAR T, and this is a 3D culture with CAR T infections, beautiful. And then, the latest, and I'm done. You know that in our brain and spine, microglia are the macrophages, or the immune cells killers of the human body. We took the neuroblastoma and glioblastoma cancer cell line, and I put together with microglia, and microglia is lowering them, killing them, but not very good. When we injected Vaccinia virus and mixed it with microglia and neuroblastoma, 50 times more killing the tumor. So eventually we could use, we did not do any human trials up to now, we could use the human glioblastoma, we do not know allogeneic or autologous will work, and infect it and target a cancer in the brain and spinal cord. So I think I am done. I just wanted to show you that we did the basic experiment, and I come back to the starting slide. 230 years ago, the drug was discovered and developed, and today, hopefully, we could repurpose this, and we will have, instead of a very successful smallpox vaccine, hopefully a successful cancer vaccine for everyone. Thank you for your attention.